All right, now here is where the reason I didn't write this hydroxyl group in, because it's going to be connected to the amino sugar here. We've pretty much done all our connecting to this number 4 carbon, right? And cellulose and all those others, it's always been 1 to 4. This is actually going to be a 1 to 3. So we're going to have the oxygen that's going to come up here, and it's going to bind right here. So this is a beta 1 to 3 linkage. So a little bit different kind of linkage than what we've been showing before. And this side here is glucouronate. Glucouronate. Or if it was protonated, it'd be glucouronic acid. Okay, so this disaccharide, heterodisaccharide, in which we have the 2 different sugars, actually becomes the repeating unit. So this whole thing repeats. And it's going to form a beta linkage to this 4 position. So the linkage between this repeating disaccharide unit here is a beta 1,4 linkage. All right, now I want to show you some variations upon this theme. But I'm not going to draw all of them. Some of the structures get pretty complex. So this is what I just drew up there. That's a hyaluronate or hyaluronic acid, depending upon its protonation status. And that's the repeating unit. So here's another example. They can also be sulfated here. And so in this case, we have the sulfate here at that number 4 position here. That would, in our other situations, that was involved in a glycosidic bond. Now it's free and it becomes sulfated. So that's going to create more of a polyanionic characteristic. And so this is known as chondroitin 4-sulfate. Let me see if I have another example here. Okay, so here's the next thing, and I'll kind of reiterate this on the board. So these things, typically, particularly in connective tissue, these glycosaminoglycans, these heteropolysaccharides, are not found by themselves, but are actually found connected to peptides. And so the way this actually works is you'll have a chain down the middle of the hyaluronic acid. So the repeating disaccharide units are repeating in there. So there'll be a long heteropolysaccharide chain. And those in turn will be attached to short peptides that come off the side of that, right? And that short peptide is known as the core protein here. Then coming off that short peptide is our other small chains of glycosaminoglycans known as keratin sulfate or chondroitin sulfate. So you have basically a mixture of these different heteropolysaccharides, these glycosaminoglycans, that are linked together by these short peptides. There's this main chain down the middle, and then the short peptide comes off, and then there's other little glycosaminoglycans that come off those peptides. And that whole structure is known as a proteoglycan. Here's another way to look at this. Here's our hyaluronic acid backbone. Here's our little peptide chain, core protein, and then here's the little short little glycosaminoglycan chains coming off that. This is, I think the composition of this, again, it's important in connective tissue and cartilage. The composition of this is why you have some claims for chondroitin, as a dietary supplement and that sort of thing. I'm not sure how validated those claims are, but people will sell it to you. All right, let me go back and just kind of write some of that stuff down. So we have the glycosaminoglycans. We talked about the structure here. Then I'll say is they can be linked to a peptide known as the core protein to form an overall structure known as a proteoglycan. Again, this is particularly important in connective tissue. All right, so that's an example. By introducing this molecule, we basically introduce a couple of different concepts. One of them is that polysaccharides don't always have to be just the same sugar in there, that you can actually have different sugars incorporated into the same polysaccharide into the polysaccharide. And often, some polysaccharides can be linked to other biological molecules such as proteins. And so this is one example. There's another one which we don't get into this class called peptidoglycans that are important in bacterial cell walls. But what we are going to talk about is glycoproteins. Okay, so glycoproteins, we spent a long time talking about protein structure, right? We talk about the peptide bond, alpha helices, the secondary structure, tertiary structure, quaternary structure, bringing it all together. All that time we were basically ignoring the fact that a major fraction of proteins also have a significant amount of carbohydrates attached to them on the surface, okay? And so if we look at the roles of glycoproteins, it's the same kind of diversity. So we can have enzymes that are glycosylated. So again, we can have enzymes. An important one is immunoglobulins. Antibodies. Now, why is this especially important? Because it has big biotechnological implications. So, as I mentioned, the business of biotechnology is to take a therapeutic protein, something like insulin, and rather purifying it from its native source to isolate the gene that encodes it, put it into an expression organism. In the case of insulin, it's E. coli. Get the E. coli to actually express it, and then it makes for an easy purification source, and you can purify and you make a nice source of insulin. One of the major classes of biotechnological processes these days are monoclonal antibodies. This includes things like Herceptin, which is treatment for breast cancer. Actually, there's a whole suite of different monoclonal antibodies that are used. And so there, it's actually the isolation technology. You basically isolate the monoclonal antibody that's also ultimately going to interfere with some aspect of the disease process. And again, you clone the gene for that monoclonal antibody. But antibodies are glycosylated, and E. coli doesn't glycosylate proteins. And so you can't just take the human gene and stick it into E. coli and get it to express, because it won't have the appropriate glycosylation pattern, it won't have the appropriate structure, et cetera, like this. You have to actually put it into a eukaryote, a mammalian cell, in order to express it. So the mammalian cells that have been adapted, there's several different lines. One of them is Chinese hamster ovary cells. This is like an experimental line. Take a hamster, ovary cells, and it's basically, a perpetual cell line that can be engineered to actually encode genes for these immunoglobulins. And then these cells, engineered cells, are grown up in big reactors, and then you isolate your protein from there, and now you have your therapeutic protein. So if you go down to Vacaville, head down 80, and just before you get into Vacaville on the right, there was what used to be actually owned by Genentech, but a great big protein production facility down there. Genentech shedded it and kind of split it off, and now it's its own company. But regardless, and what they do in there is they grow Chinese hamster ovary cells that have been engineered with particular therapeutic monoclonal antibodies, and they express them. Now, the thing about the growing Chinese hamster ovary cells is it's expensive. Growing E. coli is really cheap and easy. Growing mammalian cells is pretty expensive. You have to have these special growth factors and all that sort of stuff. David?
Sorry, just out of curiosity,
Yeah.
Is that similar to the synthetic meat process of being able to produce that sort of?
Well, yeah, so they're both.
In which it's like a giant vat that you actually have.
Yes, exactly. And I don't know exactly, I'm not that familiar with that technology, but I'm assuming that they're making muscle protein basically in those, so a similar kind of thing. Except now you're making antibodies that have some sort of therapeutic value. And so basically down there, they grow these cells. It's very expensive, and you'd much rather do it in E. coli, but the reason you can't do it in E. coli is because of the glycosylation, that you need a mammalian cell to be able to properly glycosylate the protein after it's translated. That also, the whole sort of biologic, they call those biologics, that whole development of that industry also kind of changed a little bit of the strategies in terms of making money in pharmaceuticals. What used to be said is, if you want to make money, find a drug that treats sick people. If you want to make a lot of money, find a drug that treats healthy people. And sort of the idea was, aspirin is a drug that treats generally healthy people, because your market is huge. There's a lot more healthy people than there are sick people, right? Your market is huge. That's what makes Viagra great, right? There's a big market of old guys out there that are generally healthy but could use a little bit of help in the ED department, right? So you make a lot of money. But with these monoclonal antibodies are usually directed at some pretty specific disease process. So the model actually became, okay, we're gonna make these therapeutics that treat a small fraction of people, but we're gonna charge a tremendous amount of money for it. Herceptin might have just come off patent, but before Herceptin was on patent, or is off patent, I think they were charging $70,000 for a course of Herceptin. The other kind of interesting thing about the facility over in Vacaville is, at least a few years ago, we used to take tours over there until they kind of shut things down. But up until a few years ago, at least, the last time I was aware, they would make the protein in Vacaville, and then they would ship it down to South San Francisco where it'd actually be packaged to be sent out and sold. Now the thing is, if you drive down 80, have you ever seen many trucks like that says Genentech on there that are carrying that stuff? No, you never see it. Because I think it does go down I-80 to South San Francisco, but they're probably in like these like simple white panel vans that they do at 3 o'clock in the morning, because there's millions and millions of dollars worth of product that are being shipped. They would ship them in liquid nitrogen in there. So that's just kind of an interesting kind of thing. We do occasionally get students who go and get jobs at the production facility in Vacaville, usually involving QC. You have to, if you're going to make a protein that's going to be injected in the bloodstream, you got to really make sure that it's clean and pure and all of that. So there's pretty heavy duty QC processes and that whole thing. What else it was kind of interesting about visiting there? Oh, the water supply. You need water to be able to grow these things. And so they had their own water. I don't know if they had their own wells, but they had their own water treatment facility. They couldn't just rely on the city of Vacaville water, right? The water had to be super clean and all of that, so they had their own water treatment facility for that. All right. Glycoproteins, enzymes, immunoglobulins. I'll show you an example of another one, mucins. Just because it's kind of cool. They're kind of cool looking. I'll show you a picture of them a little bit. One of the things that makes your spit kind of slimy is that there's these proteins in there called mucins. They have a whole bunch of carbohydrates, and those carbohydrates will, because of all those hydroxyl groups, will bind a lot of water molecule, hydrogen bonds with a lot of water molecule, and it ends up one of the things that makes spit kind of slimy. I guess I was always thinking about like when you spit out, slimy spit out, but most spit probably goes down the back of your throat, so it's good to slimy to help kind of lubricate your esophagus. Okay, so what do they do? What's the function of the glycoproteins? Functions of the sugar part. Function of the sugar like this. One is they might help define protein structure. So we spent a fair amount of time talking about when a polypeptide has to fold up, it has to fold up in one of what might be several potential stable structures, and that these fold up into these stable structures, and that you might have to take chaperonins and all that sort of help stabilize it. And we talked about the different hydrogen bonding and ionic interactions and hydrophobic effects that end up controlling over there. So you can basically imagine now if you have a polypeptide that has a bunch of sugars attached to it, that those sugars are going to ultimately influence the overall structure of the protein. Secondly is they'll mediate recognition events. I think I referred to this a little bit when I introduced carbohydrates. So that often proteins on the surface of a cell, it'll be integral membrane proteins. Also, those proteins on the surface of the cell will be glycosylated. So that the surface of a cell actually has a bunch of sugar residues sticking out at it. And the pattern of those sugar residues is going to differ from cell type to cell type. So one of the ways cells recognize each other is by the pattern of sugars on the surface of those glycoproteins embedded into the cell membrane. All right. We talked about some of the different functions, but I mean some of the different functions of the enzymes that are or the proteins that are glycoproteins, and then also sort of the role of the sugar within the proteins. Now let's ask the question: well, how is the sugar actually attached to the protein? Okay, so this is linkages. And there are two main classes of linkages. There are N-links to asparagine. So if we have, I'm going to draw the peptide backbone here. Here's a peptide backbone. So here's one amino acid, and this is going to be asparagine. Asparagine. I know that was like 2 weeks ago you had to memorize that or something like that. That one had CH2, C double bond O, NH2. That's asparagine. So that's our side chain. But don't draw this H2 in there quite yet, okay? Because what I'm going to do is I'm going to show you that that's actually going to end up being linked to an N-acetylglucosamine. So this is N-acetylglucosamine. So this is the sort of the first thing that happens is you take an asparagine, you add on that glucose, okay, and then most often there'll be additional sugars added to this at this 4 position, and the chain will kind of grow out from there.
Professor?
Yes.
What is IZ?
What is what?
IZ mean?
Oh, this is
The agent.
NH.
Oh, thanks.
Now the actual glycosylation process is relatively complex, so I'm just kind of giving you sort of the basics. Some of the glycosylation happens co-translationally, so basically the protein is, as it's being translated, it becomes glycosylated. Some of the glycosylation process actually involves transport through the Golgi apparatus, and then the complex sugars are added into while it's being processed through the Golgi apparatus. A complex set of sugars will be added on there. We're not going to get in for this course. We're not going to get into that detail, but we will get to the next. Other than the N-link glycans, the other way that sugars attach to proteins is through serine or threonine. Those are O-links to serine, or threonine. So I'm going to draw the backbone again here. Now I'm drawing serine, but it could also be, if that was a methyl group, it would be threonine. So this is serine if H, threonine if it's a methyl group. Because I remember the structure of the amino acids, the side chain. Serine was CH2OH, and then threonine, you basically take off one of the hydrogens and put a methyl group on there. Okay, so now the same kind of thing, except now we're going to link it to a disaccharide through an alpha linkage. Again, we have, you see this N-acetylglucosamine kind of shows up a fair amount. But now we're actually going to have another one of these beta 1,3 linkages to a galactose. So this is N-acetylglucosamine. And then this is galactose. And again, we can add additional sugars on beyond there, but this is sort of the most fundamental sugar connected to a serine or threonine is this disaccharide. All right, so I'll finish up. I'll show you a few slides here. I'll finish up with that, and then we'll take a break. We'll break until tomorrow, and we'll pick up on lipids. So here, this just kind of repeats what we did here. We can have the sugars either attached via a serine or attached via an asparagine here, the N-linked or the O-linked glycoproteins. And then here is just to illustrate the fact that if here is our membrane, plasma membrane, the cytosol is in here, so we're looking at up to the top, we're looking at the outside of the cell, that we have these integral membrane proteins. And later on, as we talk about membrane structure, we'll talk about different ways proteins are associated with membranes, but this is an example of an integrated membrane protein, in which there's a part of it is on the outside, and then the outside is going to have a bunch of sugars attached to it. And then those sugars basically, again, are unique to each cell type. That pattern of sugars are unique to each cell type, so they end up becoming sort of a barcode for that particular cell. And it mediates certain events. So, for example, if here we've got a cell here, and the cell's responding to its virus, whether it's a bacterium or to its environment, responding to its environment, whether it's a bacterium. Often, what it does, it basically recognizes the carbohydrates on the surfaces of these different other cells that it's interacting with. Okay, this is the mucin. This is the one in your spit, basically. Here's the polypeptide component, and it has this great big collection of sugar residues on there. Those sugar residues interact with water a lot, and that kind of gives what the sliminess of spit. Here's another way of looking at carbohydrates on the surface of protein. This is just a transmission electron micrograph. Here's the cell membrane, and we have all this fuzzy stuff that's coming off there. That fuzzy stuff is the carbohydrates that's sticking out from the glycoproteins embedded into that membrane. Okay, I'll leave you this final thing because glycosylation pattern of blood proteins is what's responsible for different blood types. So again, what we have is a on the surface of the proteins that go in your float around on your blood protein is a pattern of sugars, of N-linked sugars. And so we basically have the N-acetylgalactose. We basically have this pattern of sugars. Everybody has this pattern of sugars. Okay. Now some people will then add another sugar to make it to add this additional sugar at this position here, where N-acetylgalactose. Other people will add galactose. So we basically have different populations of people. Some people don't add any extra sugars. Those people have type O blood. Other people add this N-acetylgalactose onto that core unit, and they have type A blood. Now, if you have type O blood, and all of a sudden you get blood from somebody else that has this extra sugar on there, you'll recognize it as foreign and you'll generate antibodies against it, right? So a type O person can't receive blood from somebody who is type A because it's seeing this different pattern on the surface of the blood proteins, and then it recognizes that as foreign and you get an immune response. Similarly, a type O person cannot get blood from somebody who has this galactose added here, because their body will also recognize it as a foreign protein and generate an antibody. Okay, so type O people can only get blood from type O folks. Now, what if you're type A? Okay, well, if you're type A, you can actually get blood from type O because you still have this same core. Your body won't recognize the type O blood as being foreign, right? Because it still shares that same core. Okay, so if you're type A, you can get blood from type O people or from type A people. You can't get it from type B people because it has a different sugar. Your body will recognize it foreign. Similarly, if you're type B, you can get it from type O, but not from type A. Now, there's some people that actually have a mixture of both, type AB, right? And so the type AB people can actually get blood either from O or from if you're A or for B. So the type AB people are the universal. Let's see. The type O people are the universal donors because their blood anybody can handle. The type AB people can get blood from anybody else. So that's basically the nature of this blood typing system has to do with the glycosylation pattern on proteins on the surface of blood proteins.
I have a question.
Yes.
Related to this. So those people who are A and B, since they need those two specific extra sugars, do they operate, I guess, at like a relative deficit compared to people who are O who don't need those extra sugars?
What do you mean by deficit?
Like, are they expending more energy to create?
No. The amount of relative to overall metabolism,
negligible.
the amount of sugar that is involved in these things is really minuscule. So it's not going to be an energy thing. But it's more like who can you get blood, who can you donate blood to, who can you get blood against, and all that sort of thing. All right. Anybody have any other questions? All right, very good. We'll stop here. We'll pick up tomorrow morning, and we'll get into lipids.